So metabolomics is the study of small molecules called metabolites, and these are present in biological samples. And once we study and measure these metabolites, we get information about metabolic pathways. And more recently, what we found is that if we understand these metabolic pathways and how they're changing under different conditions, we start to understand disease and development of disease and health. So for example, in the core, we are working with others in applying it right through from nutrition research through to development of disease biomarkers. So we could take biological samples, in this case it could be plasma samples from somebody before an intervention, and this could be a dietary intervention or it could be a drug intervention, and then we take those biological samples at the end of the intervention. We quantitatively measure the metabolites present, both pre the intervention and post the intervention, and we're looking to see then what metabolites are changing as a result of the intervention, and then if we can map it back to metabolic pathways and have a better understanding of what the intervention is doing. We can work right through from design of the study and helping individuals design the study through to measuring the metabolites and getting quantitative information about the metabolites, right through to data analysis. And ultimately we want users to publish the results, and we'll help facilitate that as much as we can.
So my research is based on a particular subtype of breast cancer called triple-negative breast cancer. And one of the key questions around triple-negative breast cancer is trying to identify where women are likely to develop a metastasis. We're particularly focusing on tiny particles called nanoparticles. They're up to 1,000 nanometers in size. And the subtype within those that we are looking at specifically are extracellular vesicles. But in the context of metabolites, we're hoping that we will be able to generate a metabolomic signature which will align with the patient response to treatment and also may help us identify where that cancer is more likely to metastasize to. Well, as a scientist, it's very important that one engages with multidisciplinary team research. The facilities here in the Conway are second to none in relation to the accessibility to equipment to allow you have ideas, come up with hypotheses that you can then actually bring to fruition. It allows you to have ideas around things that puts you into a very competitive area. So many pathologies, you have to have a multi-pronged approach. You can't do it on one's own. So I think embracing the technology and the expertise that's here with different groups is so very important and can only be good in the future.